You're listening to Conferences Online Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Today is September 16, 2011, and I'm your host, Dr. Jay Portnoy. Our topic today, atypical infections and asthma. Our presenter is Dr. Prescott Atkinson. He's a professor and director of pediatric allergy, asthma, and immunology at the University of Alabama at Birmingham.
Good morning. I want to first thank Dr. Atkinson for being a brave soul to come on COLA. For someone who hasn't done it before, he was willing to try. A lot of people are a little concerned about how this whole process works. Hopefully it will be relatively easy, and we'll get you to give us another talk at some time in the future. This morning, for our second talk, Dr. Atkinson going to talk on atypical infections and asthma. I've known Dr. Atkinson for a long time. He and I are both program directors. He's in charge of the allergy immunology training program at University of Alabama, Birmingham. He has a marked interest in immunology and also in atypical infections and asthma, and he was willing this morning to talk to us. And I want to welcome Prescott, and thank you very much for doing this.
So welcome to Conferences Online Allergy, Dr. Atkinson. I can give you control of the keyboard and mouse, and then
okay, all right.
Mike, you can then control it, and should be all yours now.
All right, well, very good. Well, thank you very much, Paul and Jay, for the invitation. I do find this program very user-friendly. I'm quite impressed, and I really enjoyed what I heard of the last talk. It sounded very interesting indeed. I'm going to be a little more focused in my presentation than the last one, which looked more like an overview of a lot of different dermatologic conditions. I'm going to be talking about atypical infections and asthma and pretty much focusing on one organism. As you may be able to guess from the topic title, I'm really going to be talking about mostly Mycoplasma pneumoniae and evidence to suggest that it may have a role in pathogenesis and also its role, clearly has a role in acute exacerbations of asthma and issues in diagnosis and treatment. Let's see if I can change. Okay. Before I get started talking about the organism and atypical infections, and by the way, the atypical infection, when we talk about atypical bacterial pneumonias, we're usually talking about three organisms: Mycoplasma pneumoniae, chlamydia, or what is now called Chlamydophila pneumoniae, and Legionella. And Chlamydophila pneumoniae and Mycoplasma pneumoniae may have a rather similar phenotype in terms of producing exacerbations. They're clearly organisms that are designed for chronic or at least long-term infections, pretty much limited to humans. Legionella may be more of an acute infection, and although it can look rather similar, is a different type of organism. But I'm not going to really talk about either of those other two organisms. I don't really specialize in them. And as far as chlamydia or Chlamydophila pneumoniae, though, it could be viewed as a rather similar, and studies have shown that it also plays a rather, seems to play a rather similar role in exacerbations of asthma and so forth. So before I get started, I'm going to talk a little bit about the airway microbiome, some emerging information with the newer chips that are being used to look at microbial populations in different areas of the human body, as well as in various aspects of our natural surroundings. It's very powerful techniques that are just beginning to open up this area. The data very preliminary and not necessarily very clear as far as their implications yet, but they're very interesting. I thought I'd just touch on a couple of recent publications and then focus on Mycoplasma pneumoniae, some evidence related to acute exacerbations and a role in chronic asthma, problems in diagnosis and treatment, some information related to what's known about virulence factors for this organism, particularly how it may activate immunologic cells, a newly described toxin which is related to pertussis toxin, animal models of infection that support a role in chronic infection and airway hyperreactivity, And finally, an issue that could become a big problem in the near future in the U.S., emerging macrolide resistance in this organism. So first, I want to talk about a couple of papers that have come out recently, this one in 2010 by Hilty and colleagues, in which they looked at microbial populations in COPD, asthma, and normal individuals, both in adults and children. They looked at the 16S ribosomal RNA using PCR and sequenced about 1,000 clones for each individual in each group. And they came up with the observation that there appears to be a normal flora in the upper airway, or in all of the airways, and the lower airway is rather similar to the upper GI tract, with maybe about 2,000 bacterial genomes per square centimeter of airway. And the populations appear to differ in asthma and COPD, with the asthmatics and COPD patients tending to have higher diversity and higher numbers. And there's a prevalence of gram-negatives, such as Haemophilus, in patients with asthma and COPD as well, and gram-negative anaerobes, which are difficult to culture, like Prevotella, which may have been under-recognized in the past as being prevalent in normal airways.
Dr. Atkinson, that's kind of amazing information to me. I was always raised with the idea that the airways were sterile, that they didn't have a lot of bacteria in them, and now we found out that there actually is a flora?
That appears to be true, and as I say, this information it's using techniques that are opening windows that we haven't really looked in very well before, and it's clear that a lot of the organism, or it appears likely that a lot of the organisms that are being detected may be just passing through. They may be organisms that were inhaled and are being cleared by the airway, but some of them are likely actually resident and proliferating there. And all that information is, I think, what's going to need further study in the future. But it does appear that the airways are not as sterile as we were led to believe, and I'll show you what I mean here. So this is the really interesting graph of showing the diversity of different organisms that are there. And the interesting thing is that in the controls, the orange and yellow rings are the proteobacteria, or gram-negatives, like Haemophilus. And you can see that those rings are much larger in the COPD and asthma patients. And similarly, the Bacteroides type anaerobe, such as Prevotella, shown in purple, lavender, are expanded in the normals, in the inner ring as well. So there's a very different, there appear to be significant differences in the flavor of different bacteria that are being obtained through these techniques. So the second paper was a paper that came out in the JACI earlier this year, and it was a paper by Huang et al., and they used samples that were obtained from an earlier multi-institutional study published also, I think, in the JACI in the fall of 2010 by Sutherland et al. And they looked at bronchial brushings, and they used microarray as well as parallel clone sequencing analysis from 65 asthmatics and 10 controls. And they found what appears to be a substantially higher microbial diversity in the asthmatics. And this seemed to correlate with airway hyperresponsiveness and also response to antibiotic therapy. So these were observations that were not published in the original study, and this study was not designed for this purpose, but they're utilizing samples that were obtained in this study and then reanalyzing them. This is a little cladogram showing the incredible diversity of bacteria that they're detecting in the airways. And I think it's probably highly likely that many of these are just present but not actually replicating. They're things that the person has inhaled. We know that airways in asthmatics and COPD patients have more mucus, and that the ciliary clearance may not be very good. This may be kind of a reflection of the possibility that the airways are just dirtier there. There are more bacterial products, but not necessarily telling us that the airways that there are actually all these organisms growing there. I don't think that's likely to turn out to be true. But one thing, and this shows this shows the amount of bacterial products that could be expanded using PCR in 47 asthmatics compared to 5 controls, and a pretty significant looking difference there. And again, just telling you that the airways in a lot of asthmatics appear to be dirtier than in control individuals. The numbers of controls are relatively small, analyzed here. And then finally, because this original study was a treatment trial, they did have information related to patient responses and to a trial of clarithromycin. And although again the numbers here are pretty small, it did appear that patients with higher bacterial diversity did exhibit a response to clarithromycin treatment compared to patients with lower bacterial diversity in the airway. So the summary that you could say from these still rather preliminary studies that are rather similar to each other, but definitely need more studies to replicate them and expand their findings, seem to imply that there is a normal airway flora, that there are bacteria that are commonly present there. They may not be easily culturable, but that normal flora may be deranged in people with asthma and COPD. There appears to be higher bacterial diversity in many patients with asthma and COPD. Some of those may just be, as I mentioned, they may just be contaminating the airways in people who don't have very good mucociliary clearance and have increased mucus in the airways. But there does appear, at least in a subset of patients, to be a correlation of improvement with antibiotic treatment and a correlation with airway hyperreactivity. It's a complicated subject, and I think the data are still preliminary, but they're intriguing and do suggest that there is that just as airway allergic disease clearly has a role in asthma pathogenesis, that bacterial products and actual bacterial flora may also have a role as well. And that sort of takes us into the topic related to the main subject that I wanted to talk about today, which was Mycoplasma pneumoniae, for which we have a fair amount of data, but still waiting for more conclusive information. So asthma, as everyone here doesn't need reminding, it's a chronic inflammatory disease. The essential characteristic is that it's reversible, that it results in airway hyperreactivity that is at least to some degree reversible. You could ask the question: Is asthma today the same disease that physicians were treating early in the 20th century? Our predecessors didn't seem to have a very high opinion of asthma as far as a severe disease. Sir William Osler said that the asthmatic pants into old age. Oliver Wendell Holmes, who was an asthmatic himself, said that it was a slight ailment that prolongs longevity. Of course, at that time, physicians were struggling with a whole variety of much more severe infectious diseases in Western countries, and those have been at least temporarily tamed with the advent of antibiotics. So it may be all a matter of degree. However, we know that asthma today is a big problem, very expensive problem, affecting over 24 million people in the United States, disproportionately higher in certain ethnic groups and in certain socioeconomic groups, and is a substantial cause of mortality every year. These people are not wheezing into old age. There are people who are dying from acute asthma attacks across the country. So it is a big problem, and there may be, what we'll look at now is the possibility that some of this problem may be related to infection, or at least to bacterial products. Asthma pathogenesis, there are many, many different aspects, of course, in the literature. There's a clear genetic predisposition, which we've known about for a long time. And genetic studies have revealed a number of genetic loci and some gene polymorphisms. And many of these appear to be things that should be related to host defense and inflammation. Among environmental influences, we know that allergen exposure is very important. The majority of childhood asthmatics have airway allergies. Irritants such as diesel exhaust, tobacco smoke, other pollutants also have a proven role. And infection and microbial products are probably also important. There's a lot of information, particularly from the group in Wisconsin on viral infections in early childhood. The hygiene hypothesis, which seems to show that bacterial products, exposure to bacterial products can actually have a protective role. And then the role still unclear, but I hope to show a little later in this presentation that stronger data showing that atypical bacteria such as Mycoplasma pneumoniae and Chlamydophila pneumoniae may have a role both in pathogenesis as well as in acute exacerbations of asthma, and there's something that you should maybe think about. So in asthma pathogenesis, we're thinking about genetic, genes, environment, maybe psychological factors, stress. Osler believed that asthma was largely a psychological problem, that it was largely a psychiatric issue. We don't think that today, but clearly stress does play a role in exacerbations. I want to digress for just a second and talk about something that the older physicians here probably remember. Back in the days when I was a medical student, here's a quote from Harrison's in 1983, talking about a disease that we still see very frequently, peptic ulcer disease, and the etiology was unknown, and they were looking at a number of different aspects related to peptic ulcer disease. This is what Harrison said. It looked like there's an unfavorable balance between gastric acid peptic secretion and the mucosal resistance to acid. So they were looking at all kinds of things, frequencies of blood group antigens, increased secretion of digestive enzymes, environmental risk factors such as tobacco smoking and lots of coffee, the type A personality that I can remember one of my teachers putting up on the board when I was a medical student talking about it. And then, of course, today we know that peptic ulcer disease is largely an infection with one organism, usually caused by Helicobacter pylori, for which Nobel Prizes were awarded in 2005. Asthma is not going to turn out to be such a simple disease, certainly, but it does tell you that chronic infections can cause chronic inflammatory processes that can cause disease in a small percentage of patients affected. I mean, peptic ulcer disease was difficult to ferret out, but it's a common infection affecting perhaps 50% of the human population, but only a small number of those patients are affected. Genetics are likely to play a role in determining host susceptibility to this organism and how much havoc it can wreak in the intestinal or the gastric mucosa. So now I'm going to talk about the main type of atypical infection that I wanted to focus on today, which is Mycoplasma pneumoniae. It's actually one of about 17 species that have been isolated from humans. 5 of them are known to be pathogenic. Mycoplasma pneumoniae, first called Eaton agent, was first thought to be a virus because it's so small that it's actually filterable through the smallest types of filters that really distinguished viruses from bacteria back in the 40s and 50s. Then later found to be an unusual type of bacteria, very difficult to culture, but we've eventually developed very highly enriched culture media so that it can be grown in pure culture. It typically infects ciliated epithelial cells in the upper respiratory tract and oral cavity initially, and then moves into the lower respiratory tract frequently. But the infections can be very pleomorphic. It has to be able to adhere to epithelial cells to cause disease. Mutations that knock out this ability to adhere eliminate pathogenicity in animal models. It's a very tiny organism, as I mentioned, one of the smallest types of bacteria known, down to about 1/10 of a micron. Unlike other bacteria, mycoplasmas don't have any cell wall, and they also are unique in that their plasma membranes contain cholesterol. They have a tiny genome. It's been stripped down to just the bare essentials, only 677 genes compared to over 4,000 for E. coli. And this is a process that's called reductive evolution, and it is the hallmark an intracellular parasite or an intracellular bacterium, but an intracellular role for this organism is not well established. What is known, though, is that it's kicked out most of the genes that it needs for normal existence, such as the ability to synthesize purines, most amino acids, and even cholesterol, all of which it needs to exist. So it basically is essentially a parasite. Cannot grow on your potato salad or anything like that out in the world. It has to infect a host organism.
Yeah, one thought that just popped into my head, is this the way that mitochondrial started out?
Exactly. I mean, that process, of course, has probably occurred multiple times. There are lots of another area of intense interest in among microbiologists right now is the finding that a lot of organisms have commensal intracellular organisms that are growing within them. Algae, for example, frequently in things like giant clams. They're commensal organisms that are really essential for the host to survive. And mitochondria and chloroplasts, presumably eons ago, took up such an existence and became integral to our cells and essential for our very existence. These guys are not at that point yet. They're still troublemakers in general. But they do appear to have reached a point where they're just like the chlamydia, for which this is well established, who are obligate intracellular parasites. Mycoplasmas may be facultative intracellular parasites. They may have an intracellular role or an intracellular phase in their existence, and the importance of intracellular infections in other bacteria as well as the mycoplasmas and others, that they may be more resistant to antibiotic treatment in those when they're in that particular phase. They may not be replicating. They may not be very biosynthetically active. We still have a lot to learn to know whether that is true or not, but there is some in vitro data that shows that Mycoplasma pneumoniae can move into an intracellular environment within cells from time to time. Here's what the organism looks like. This was a picture that a graduate student in my lab took some years ago by electron microscopy. It's got this peculiar flask-shaped appearance with an elongated adherent tip that's studded with adhesins. It's like and it's also motile. It crawls around on the plate. You can see videos of the organism crawling around, and it needs this adhesive tip to do the crawling as well as adherence. And it also uses it. It's also essential for proper cell division. So it's a specialized little guy. It's got doesn't have a lot of genes, but actually the majority of them we're not sure of the function yet. There are many of those genes that are not, the function of which is not clear. Very high. You could think of it as a highly sophisticated organism. So what do the clinical manifestations of these infections with this organism look like? They're actually pretty pleomorphic. The classic is a bronchopneumonia, but they can also just produce a mild upper respiratory set of symptoms during acute infection, or maybe very little symptoms. Family studies, largely done in the 60s and 70s, did not show that if you went to the family of an index case with an atypical pneumonia, many of the other family members were also infected and might really not have had a lot of symptoms. The symptoms may be flu-like. They may be accompanied with high fever and chills. There may be a pharyngitis and some hoarseness, nasal congestion with coryza. This can progress to wheezing, pleuritic chest pain, and the typical tracheobronchitis or atypical pneumonia. Reinfections are common in individuals. It's a very common infection in the U.S. Perhaps 30% of community-acquired pneumonias, 2 million infections estimated per year. It tends to occur in little epidemics every 3 to 7 years or so for reasons no one's quite sure about. But it is certainly endemic in many areas, and infections can occur at any time of the year. Infections occur at all ages, but most of the more severe infections tend to be older children and young adults, adolescents and young adults. The incubation period, the onset of this type of infection is usually rather slow, and can occur over 2 to 3 weeks of gradually increasing cough, and it's also a rather prolonged infection with cough that can last for weeks, and the organism can actually be cultured from individuals even after antibiotic treatment for months afterwards in many cases. It's spread by droplets, and studies back in the 60s and 70s in the military showed that people who had their bunk in bunk rooms where an epidemic was occurring, people who had their bunk closest to the shower were most likely to get infected, probably because of the higher humidity in that area. And of course, it does occur in outbreaks and little mini epidemics regionally. The radiographic findings often may look more severe than the patient's clinical condition would suggest. Sometimes family members who were more or less asymptomatic without much fever or and minimal cough would have an x-ray and actually find a streaky infiltrate. It's frequently sort of a basilar or bibasilar streaky sort of infiltrate. There may be pleural effusions that are located or that are unilateral or perhaps even bilateral in in a significant percentage of cases. Here's a series of studies that I reviewed over the past 10 years of patients with asthma, looking at the occurrence of Mycoplasma pneumoniae as detected by PCR, and I'll go into why I basically restricted this to PCR in a little bit. Excuse me. And you can see that the positivity is, they're all less than 50% of these individuals that the organism was detected, but there's a very wide range, all the way from a minimal of 4.5% all the way up to almost 50% in one study. So it probably varies. It may vary regionally, and there's also difficulties in diagnosis, which can depend on the particular PCR reaction that's being used, how efficient it is, and so forth, the collection procedures. So point-of-care diagnosis, it continues to be a big problem with this infection, but it does appear to be capable of causing, or at least being associated with, a significant proportion of asthma exacerbations. Most of these studies were done in kids, as you can see. The other question that comes up with Mycoplasma pneumoniae is, could it have a role in chronic stable asthma? These are two relatively recent studies, or really three, that I thought I would mention. These have been largely, the most important work have been carried out by Monica Kraft and Richard Martin and their colleagues at the University of Colorado. Dr. Kraft is now at Duke University. But the intriguing study that was initially published showed that in chronic adult asthmatics, that if you looked at BAL or endobronchial biopsies by PCR, you could find the organism present there in a substantial proportion, about almost 50% of individuals, compared to only a minority of control individuals. And the numbers of controls were relatively low. So it's possible that this may not be truly a reflective of a difference in occurrence, but it certainly does suggest that the organism's there in substantial amounts, at least in some studies in chronic asthma. Also, several individuals were positive for Chlamydophila pneumoniae, another organism that could cause a very similar type of phenotype. And interestingly, the same group then did a 6-week trial of macrolide treatment in a follow-up study and demonstrated a clinically significant response only in the PCR positive group, about, I think, 0.4 liters improvement overall in that group. And importantly, none of these chronic stable asthmatics were serologically positive. They might have some antibody, but it was not really, since this is a type of antibody that's present in the normal population, there was not really a difference, a significant difference between the background in these individuals. In a more recent study, multi-institutional study, this is the study that was used in the earlier paper that I mentioned by Huang et al. Sutherland, Rand Sutherland, who's a former pulmonary fellow and now, I think, a faculty member at the University of Colorado, showed that that the organism was still there in substantial numbers, but they were substantially less than the previous study. They were trying to use this in 92 chronic adult asthmatics, trying to, again, replicate this finding of improvement with macrolide treatment. They did not get enough positives to be able to show whether this is whether macrolides showed efficacy or not. But again, none of these individuals were serologically positive, and the organism was present in a substantial proportion of the asthmatics. So the question is then, what is it doing there? Does it have a role in pathogenesis, along with the other aspects of asthma pathogenesis that we've already mentioned? The jury is still out, but it certainly does appear to be there and be capable potentially of causing, of having a role in pathogenesis and potentially periodic flares causing exacerbations of asthma as well. Mouse models of asthma using MDMA also suggests that this organism is capable of sticking around for a long time. This is a paper by Hardy et al., who showed that this organism could be instilled into mouse airways and could hang around for a long time. This is a pretty old mouse, and they were still able to culture and PCR the organism in a substantial proportion of animals pretty far out. And if you looked at airway hyperreactivity using whole-body plethysmography, there was a strong tendency for the infected animals to have higher baseline airway wheezing. I guess you could call this enhanced pause that's measured, as well as increased reactivity to methacholine. Virulence factors that the organism expresses have been, a number of different virulence factors have been described that can affect its ability to activate the immune system, cause inflammation, and potentially replicate some of the features of asthma. We know that the organism expresses adhesins on this elongated tip that I showed you earlier, and there are a number of proteins that are involved, particularly a protein called P1 that binds to sialoglycoproteins. Now these sialic acids decorate most mammalian surface proteins have sialic acid as part of the carbohydrate carbohydrate portions of surface proteins, and this organism binds rather promiscuously to these sialic acid residues. So it can actually bind to avian tracheal epithelium as well as mammalian, mouse, human, although it is not known to cause natural infection outside of humans. And potentially can cause cellular activation by binding to these surface molecules. And I'll show you a little bit of information in a second. It's been known from the 60s that this organism produces a lot of hydrogen peroxide. So you can let your imagination tell you what would happen if you had a small organism bound to your epithelial cells producing a lot of hydrogen peroxide sort of as a point source. It can actually cause hemolysis of red cells. And this hydrogen peroxide production has also been shown to be a virulence factor. Mutants which did not produce hydrogen peroxide have much lower virulence. It produces lipopeptides, which are capable of activating toll-like receptors on macrophages and other immune cells. And the most interesting thing recently is the recognition that this organism produces a toxin, which is very similar to pertussis toxin and other AB-type toxins that has enzymatic activity and has a definite toxicity for the respiratory epithelium. And we're still getting more information about this. A lot of this information has been, or virtually all of this information has been produced by one group at the University of Texas in San Antonio with a very senior microbiologist named Joel Baseman, it's certainly very intriguing, and there are a number of recent publications that suggest that it may have a role in not only in asthma but other types of respiratory problems such as ventilator-associated pneumonia. This is a ribonuclease protection assay showing that Mycoplasma pneumonia on rodent mast cells produces cytokine activation in a very similar way to what's seen with IgE in the first column. So all these little bands are just the RNA molecules for these different cytokines. So it seems to upregulate cytokine mRNA in mast cells. If you used antibodies against the P1 adhesin, you could inhibit that process. If you treated the mast cells with neuraminidase, which removes the sialic acid residues, you also inhibit that process. So the interpretation from this is that the organism's binding to sialoglycoproteins on the surface, likely, some of which would be likely the heavily sialated IgE receptor, and activating them. And this you could think of as just a model for how it might activate a lot of different immunologic cells, and perhaps epithelial cells as well, because it does have this kind of nonspecific capability of binding to sialoglycoproteins on cell surfaces. In this last paper published by a postdoc working in my lab, she created transfected cell lines of this mast cell line in which the alpha chain of the IgE receptor was knocked down using RNAi, and it completely eliminated the response, the production of IL-4 protein in secretion. So it looks like in this model that the organism's binding to the IgE receptor and triggering cellular activation through that mechanism. So that's one way the organism could produce activation. I've already mentioned the hydrogen peroxide and lipopeptides, for which there's also experimental evidence showing an important role in pathogenesis. But I wanted to talk just for a little bit about this new toxin, the CARDS toxin, because it's quite interesting. It was first described in 2006 by Cannon and Baseman as an ADP ribosylating and vacuolating cytotoxin, similar to the pertussis toxin. And they've had a number of publications since that time. Most recently one earlier this year in Chest by Jay Peters and working with Joel Baseman's group. And in this prospective observational study, they looked at 64 asthmatics who had fairly refractory asthma. And they looked at nasal lavage fluid and sputum, and used PCR and also an antigen capture assay for the CARDS toxin, as well as serology. And they found that about 50% of these chronic, rather refractory patients were positive on their initial visit for the organism compared to other controls, and that many of them, or almost all of them, were positive over a very long period of time as they were being monitored. They did find that they could detect an antibody to the CARDS toxin itself, an IgM antibody, in the positive patients, but they did not find any difference in IgG titers in these patients from other patients. So it may depend on what antigen you're looking for when you're looking for antibody. Overall, the antibody, and what isotype of antibody you're looking for in order to identify these types of patients, or to identify patients who have infections.
Dr. Atkinson, are there environmental sources of the CARDS toxin that aren't necessarily endogenous from the bacteria?
No, as far as I know, this is a specific toxin that's secreted by this organism. And as I mentioned, this organism is not like aflatoxin. Fungal aflatoxins that are made by organisms growing in contaminated agricultural products. I mean, this organism is pretty much restricted to growing in its eukaryotic host, which, as far as is known, is its natural host. Only natural host is humans. So and what I'm going to show you now is a couple of other things. which may help to kind of answer that question. Intratracheal installation of this toxin, recombinant toxin, can replicate a lot of the effects of infection with the organism in mice and also in a primate model. They found that actually the organism doesn't secrete the toxin except in vivo. When you grow it in vitro in this enriched medium, it doesn't seem to produce a lot of the toxin. This is all normalized per M. pneumoniae genome, but it produces a whole lot more of the toxin. So there may be some sort of environmental cues that the organism gets when it's growing on an epithelial surface in an animal that triggers the production of this toxin. We're still trying to understand that. This organism doesn't have a lot of well understood gene regulation, and so it's still not clear exactly what is triggering this difference in toxin production. But there does appear to be upregulated toxin secretion when it's present on an actual epithelial surface. This is a picture of what the mouse bronchial epithelium looks like with heat inactivated toxin on the left, and after installation of 44 picomoles of toxin on the right side. You can see tremendous vacuolization of the epithelia there, and also as well as a leukocytic infiltrate, largely lymphocytic infiltrate in the surrounding lung parenchyma. And this is also in the same paper, in a recent publication by the same group. They used normal human bronchial epithelial cells grown in air-liquid interface cultures. This is a really sophisticated culture technique for these cells, and these cells are stained here with 3 different stains: DAPI, which stains DNA; phalloidin, which I think is a stain for actin; an antibody to the toxin itself in red, and then the last one is all 3 merged. And the most interesting, actually, part of this to me, if you look at the bottom where they're actually looking at slices as seen from the side, you can see the little organisms along the surface, along the cell surface here in blue on the left side. And then you can see the organism, you can see the toxin also clustered on the surface, but you can also see it within the cell. And this is then seen again in the merged picture. And they have actually they have other publications showing that this toxin is entering the cell, and presumably because of its enzymatic activity, it's modifying substrates within the cell and altering cellular functions. Exactly what it's ribosylating and what functions it's interfering with or modifying have yet to be figured out. So I wanted to talk for a second about the diagnosis of M. pneumoniae infections in your asthmatic patients who are in the hospital, sick, with maybe an oxygen requirement, not doing so well. How would you want to look at them to try to figure out if they might have a concurrent M. pneumoniae infection and whether treatment might be indicated? As I'll show in just a little bit, you can't just throw a macrolide into the mix and assume that the patient is being adequately treated anymore. But I will say that serology it's not clearly reliable for point-of-care use. As I've shown before, a lot of the studies are showing that the organism may be there, and there may not be positive antibodies. This is more the case in patients with chronic infections probably than someone who's got an acute bronchopneumonia. Probably someone who's got a real hot bronchopneumonia with fever is going to have antibody that's going to develop in the usual time course. But it does take time to develop antibodies, particularly IgG antibodies. You may have IgM antibodies within 2 or 3 days, but IgG antibodies are going to take a little longer to develop, and you may not have the luxury of that much time. Culture is really out of the question. It can take up to 6 weeks. It's rather insensitive. You have to get viable organisms. They have to be transported to the lab, and they have to be cultured. And even with all optimal conditions, it can take up to 6 weeks. So for point-of-care purposes, it's pretty useless. PCR has a lot of attractions. So Here's just a little note on culture. These are what they look like under the microscope. The organism grows so slowly that actually to look for it growing in culture, you actually have to look under a microscope at the plates before you can see them in most cases. As I mentioned, it's pretty insensitive, but it is the only way to perform formal antibiotic sensitivity testing. We do know some genetic changes that are present in the organism that can tell you that it's resistant to macrolides, but for example, fluoroquinolone resistance is potentially possible, and tetracycline resistance, which is occurring in other species of macrolides, hasn't been seen naturally in pneumonia yet that I know of, but it's theoretically possible, and the only way you'll probably pick it up, the only way you could practically speaking that you could pick it up would be to do culture and sensitivity testing, which can really only be done in a small number of centers. So this is a little study that we published a couple of years ago showing that in these chronic stable asthmatic children, some of whom actually had acute infections at the time they were sampled, PCR positivity in the majority of cases, both in the controls and in the asthmatics, was not associated with any detectable antibody that was outside the range of the normal pooled serum that we were using for our positive control. So they may not have a lot of antibody. At least you can't really count on it. Everyone knows that PCR is a very sensitive technique. It's theoretically capable of picking up one genome of pneumonia in an aliquot that's used in the PCR reaction. You can detect it in asymptomatic people who aren't necessarily who don't necessarily have a lot of inflammatory responses. It doesn't require viable organisms. It will be positive when the organisms are present before the serological response can occur. It's pretty rapid. You only need one specimen. You don't need acute and convalescent samples. The disadvantages are well known. It's very susceptible to contamination. The labs have to be rigorously run with rigorous controls, but these are problems that can be overcome, and you can't do traditional antibiotic susceptibility testing with it. However, we do know that M. pneumoniae is becoming resistant to macrolides, and it only takes one mutation in the 23S ribosomal RNA gene to one base change can produce essentially complete resistance to macrolides. This is now widespread in Asia. We know it's occurring in the U.S. and Europe, but its current real prevalence in the West is really unknown, probably varies from region to region. We think this is going to become a real therapeutic problem in the not too distant future, particularly when you're thinking about treating young children. So these are some recent papers. 90 of 100 isolates obtained in Shanghai, and we have there are published papers also showing a very similar rate in Beijing in Mycoplasma pneumoniae were resistant, high grade resistance to macrolides. It's occurring in Italy at a substantial proportion, in France, and this is a paper from 2008 from the CDC, in which they were looking at an encephalitis outbreak in Rhode Island that was associated with a Mycoplasma pneumoniae outbreak, and that's an entirely different aspect. Some of the extrapulmonary aspects of acute Mycoplasma pneumoniae infection are also pretty significant. But they found almost a third of the isolates were macrolide resistant. So we know it's here in the U.S. It may be a bigger problem soon because macrolides are very frequently used, and we know that actually macrolide resistance can occur even during treatment in the same patient. That's been documented in the past. It does appear that macrolide resistant that patients with infections with macrolide resistant M. pneumoniae are a little sicker, although this is hard to tell. Many of the patients, as I mentioned, don't have a lot of symptoms. They may not be very sick. It's got a very wide range of severity. So these types of studies are really hard to do with this, but it does appear that they stay sicker longer. They don't respond to the treatment, and so they tend to have a more prolonged illness. And that patients who have macrolide-resistant M. pneumoniae tend to have antibiotic substitutions because they're not responding to therapy. So here are the 4 types of antibiotics that you could think about using for an M. pneumoniae infection. And for the pediatricians in the group, you could probably answer this question pretty easily. Which antibiotic would you be comfortable with using in your child who's less than 8 years of age? It's really only macrolides.
Yeah.
So what's going to happen if we reach the 90% or 95% macrolide resistance that we're seeing in China now, in the U.S. in the next 10 years? We're going to have an untreatable reservoir of young patients who may serve as a source of infection for grandma or their older siblings and so forth. So far, tetracycline and fluoroquinolone resistance has not really been seen to occur naturally, but it does occur in other organisms and has been induced in laboratory strains. So it's conceivable that those could be a problem also. But these are not drugs that anybody would feel comfortable with using at least as a first choice in someone in a young child. We published a paper recently showing 2 patients. Only one of these had some wheezing in the past, so these are not really asthmatics, but they were 2 children who were hospitalized with pretty severe lower respiratory infection. They were poorly responsive to azithromycin and then were subsequently found to have antibiotic-resistant M. pneumoniae, which had the same mutation. So it is occurring, and we're expecting that this rate is going to increase.
You closed our window.
How do I open it again? Let's see.
Got it?
Oh, there you are. Okay.
There you go. You want to advance one?
I'm trying to advance one. I'm not having a lot of luck here.
Let me do it for you.
Okay. This is just a picture of the PCR reaction showing that it's very easy to tell actually with the macrolide resistant strains that have been mutations that have been identified. They have different melting temperatures. This is the temperature, so the PCR product melts at a different temperature. So it's pretty easy to tell if you're dealing with a macrolide resistant strain. You can do that with PCR. If you're not going to be able to detect unknown resistant mutations, but you can look for the ones that are known, and there are only 3 or 4 really that are known at this time. And I'm about out of time here, so I know we need to finish up. We did have a patient recently who was a transplant patient on multiple immunosuppressive medications who was hospitalized with extremely severe respiratory infection, who had turned out to have co-infection with H1N1 and a macrolide-resistant M. pneumoniae. And it was only after he got started on a fluoroquinolone that he turned around. He nearly wound up on a ventilator, and he did go home on oxygen. He had a very severe prolonged respiratory infection. So this, to close, the take-home points that I would suggest that you think about, at least for your asthmatics. Well, first of all, I didn't mention, but in the first few slides, the developing data that's going to be very interesting, and I suspect there's going to be a lot of new and very interesting findings in the next several years, looking at the microbiome of patients with chronic respiratory diseases, particularly asthma and COPD, that are likely to show big differences in the microbiome and between those patients and normals. Now, what the actual meaning of those differences are is another question. It may take a long time to sort out. But Mycoplasma pneumoniae prevalence does appear to be increased in patients with asthma, at least in some studies. It's certainly fairly common in the population. We can certainly say that. Acute diagnosis of active Mycoplasma pneumoniae infection at present is best done using PCR and respiratory secretions. Sputum is probably better than anything, but nasopharyngeal swabs or throat swabs are also adequate. And macrolide resistance is emerging in the U.S. It's here. We don't know what the frequency is. It's probably fairly low right now, maybe 10, 20, or possibly 30% in some areas. But it may become a worse. It may become a more severe problem as the years over the next decade or so. And as I mentioned, it can occur even with treatment of the same patient. So you might keep that in mind for your hospitalized patients. And I think that's my last slide. If I have any questions, I'll be glad to answer them if we have time.
Okay, go ahead.
Dr. Atkins, what do you recommend, because it's not something we typically address with our asthma patients, like, their mycoplasma status, even if they're severe or admitted to the hospital, it's not something we typically screen for or treat. What's your recommendation as far as what to do when, do we screen everyone with asthma, or do you do just, acutely ill patients in the hospital, or how do you recommend we take that information?
It kind of depends on the severity of illness, I guess. Certainly if the patient has any fever, we start thinking more about, in fact, if the patient has an oxygen requirement, if the patient has, I would try to look for any, the possibility that there might be a respiratory infection that's triggering the patient's illness, and PCR on sputum or a throat swab or a nasopharyngeal swab for mycoplasma pneumonia, as well as respiratory viruses, would be something that you could think about doing if you have that available at your institution. I'm expecting, or everybody in the microbiology area is expecting and hoping that better point-of-care molecular diagnostic techniques are going to become available in the next decade. We're not there yet by any means. It's still pretty arduous, but most large centers have, or at least where our training programs are located, do have the facility to do those types of things. And if your microbiology department doesn't offer a PCR, those specimens can be shipped overnight to another center where they can be done pretty expeditiously. It's quite possible to do these within 24 hours if you really lean on the lab. Does that answer your question?
Yeah, thank you.
I assume the sample you need is like sputum, because you don't have to
any
be
type
around everybody to get it, do you?
Any type of any type of respiratory secretions. If you're really talking about an acute exacerbation, the organism should be there. So I would, you may not find it in the oropharynx as readily, but I don't know that anybody can really tell you. But certainly, if it's not detectable in sputum, you'd argue that it's probably not a real. It might be there, but it's certainly not there in quantity.
You've got my mind spinning with all of these new ideas, and I'm particularly intrigued about this CART toxin. If we can't kill the bacteria with macrolides, any possibility of making a toxoid and immunizing to the CART toxin and using that as a treatment, sort of the way we do with tetanus toxin?
You're talking about vaccines, and I really think that is the future for this organism. It's such a common infection. There were some abortive attempts, actually some pretty large attempts back in the 60s and 70s to develop vaccines. Those were done with whole organism. Now we're in the age of subunit vaccines, and this would certainly be that, and the P1 protein would be certainly very good targets for a vaccine. And I agree completely. I think that would be those would be things that would be potentially very useful. It does appear that asthmatics in particular don't seem to make humoral antibody responses as well. And I didn't mention this, but in the little paper that we published a couple of years ago in asthma proceedings, in allergy proceedings, the one significant thing that popped out from our studies was that the asthmatics didn't make that we found a lot more IgG, elevated IgG to pneumonia among the controls than we did among the asthmatics. So there may be kind of a blunted response to the organism among the asthmatics, and a vaccine might be something that might be able to boost their response and allow them to at least control the infection better.
Especially now that we have adjuvants that we can use to enhance immune responses and so on.
Exactly.
Prescott, this is Paul. I have a question if you have some time still.
Yeah.
I had actually heard Dr. Martin speak at the Aspen conference back in, I think it must have been 2002, because it was pretty fresh information at the time. And he talked about a number of these patients that had severe asthma. He was dealing with adults who basically were on every medicine at that point known to man. This is before Xolair, and just weren't getting better. And and he talks about doing PCR and thinking about mycoplasma and these people having chronic infections. Just in that study that you mentioned, they showed that in that group of patients, and he was telling that he was kind of doing it almost routinely at that point, that they were putting him on azithromycin as almost like a controller medicine with a daily dose sort of stuff. That I think he initially treated them for the infection, then he kept them on like a maintenance dose, and was able, he said, the ASPA conference showed data that a lot of these patients were able to come off lower their dose or inhaled steroids, get rid of some of the other medicines they were on, and had a better quality of life. And I remember at the time having some bad asthmatics that were teenagers here that we kind of tried the same thing, and they actually got better. My thought was that part of it was because of the macrolides, sort of like the old Teo Medrol phenomena, where we used to think that the macrolides had some anti-inflammatory effect. Do you think there's any role for some of these severe asthmatics that just aren't responding to anything, that may have a chronic mycoplasma infection, that having them on low dose antibiotic may be of some help even for a period of time to kind of get them over the hump?
Well, that is a really interesting subject, and that was the purpose of this Sutherland study that came out that unfortunately didn't have I think the one finding that they had was that symptomatically macrolides improve. Overall, there was a significant, relatively mild but significant increase in symptoms, although they couldn't prove, they didn't have the numbers of M-pneumonia positive individuals. But if you look at some of the graphs, I didn't say this, but there's a pretty interesting looking trends among the graphs in that paper related to the M-pneumonia positive individuals that were treated. But at any rate, there's at least 3 randomized controlled trials in cystic fibrosis with azithromycin. You're probably aware that a lot of the more severe cystic fibrosis patients are being treated with prophylactic azithromycin, and it clearly works in a subgroup of patients with cystic fibrosis to stabilize or maybe even improve their airway function. Now we know cystic fibrosis is a disease where microbial in which microbial infection is a big player. There's not really any data that pneumonia has anything to do with it, and the party line, I think, among pulmonologists that they are assuming that this response in these individuals is due to more to anti-inflammatory effects, which are clearly there in the macrolides. They do have anti-inflammatory effects. My question would be, well, if they're anti-inflammatory, then why doesn't everybody respond? Why is it a subgroup that's responding? But I can say that having talked to Dr. Martin, I was actually involved in a program project with him on this topic. He actually got interested in this topic because he had a patient who had been sent to them with highly refractory asthma, and they did EM on bronchial epithelial cells and saw the mycoplasma on the epithelial surface, and they put the patient on clarithromycin, and the patient got dramatically better. And then it turned out that every time they took the patient off the clarithromycin, he got worse again. So he stayed on clarithromycin at least for a decade. So it might be worth a try if you have a problem patient who's really not doing well and he's on everything and you can't seem to getting better. I would certainly think that a prolonged trial of macrolide and maybe then macrolide prophylaxis with macrolides might be worth a try. And my guess is that just like everything else with humans, it's probably not going to work for everybody, but it might work. You could try diagnostic studies, looking to try to see whether the organism is there. A lot of these patients are really not suitable for bronchoalveolar lavage or things like that, but if you can get a sputum sample or something like that, you can try to see if there's any signs of PCR positivity and that sort of thing. But even if it were there, proving causality is another issue. There's clearly a lot of bacteria, or there appear to be a lot of bacteria in the airways in these types of patients, and proving causality is always going to be the big issue.
Well, a lot to learn. It sounds like a very exciting field, and we're hoping that you'll come back maybe next year and update us on what you're finding out.
No, I appreciate the invitation. It was a lot of fun. Thank you.
Thank you. We'd like to thank Dr. Prescott Atkinson for his review of atypical infections and asthma. That's all the time we have now. We're going to stop at this point. This has been Conferences on Lyme Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Join us next week for our continuation of our immunology series. We'll also meet with Dr. John Lantos on ethics and pediatrics. In the meantime, have a great weekend, everyone, and we'll see you next time. Thank you.
Thank you.
This has been an ACAAI production. To learn more about Conferences on Lyme Allergy or the American College of Allergy, Asthma, and Immunology, go to www.acaai.org. See you next time.